Assalamu alaikum everyone, this is Abdullah Khalil and this is the platform where you learn O-level, A-level and IGCSE biology of various UK examination boards. Now we have started energy and respiration from A2 biology and we have already completed the first part of this video. If you haven't seen that, make sure to watch that first. We have already discussed the use of energy, the need of energy in the living organisms, and how ATP is the most suitable candidate to provide energy for all the energy requiring processes in the cell. We have already discussed the stages of cellular respiration, glycolysis, link reaction, Krebs cycle, and oxidative phosphorylation. In today's class, first we are going to look at the summary of all these processes, and then we are going to discuss what happens if oxygen is not available for the cell. So if we look at all of these processes together in a diagram, it would look something like this. We can see that glycolysis is happening here outside the mitochondria in the cytoplasm. Glucose is broken down into pyruvate during glycolysis. Once the pyruvate is formed, it enters the mitochondria. In the mitochondrial matrix, the second reaction, link reaction, happens. There is no ATP formed during link reaction. The end product is acetyl coenzyme A. During all these processes, hydrogens are being released. During link reaction and Krebs cycle, carbon dioxide and hydrogen are being released. All the hydrogen, now carbon dioxide is the product of respiration. It is released outside the cell. But hydrogens, the hydrogens that are released from the glucose are actually accepted by the acceptor molecules NAD and FAD. We have already discussed that. During Krebs cycle, again, ATP is being formed and hydrogens are being released. Carbon dioxide is also being released. Carbon dioxide that is released will go out of the cell. It's the product. But the reduced NAD and reduced FAD, they have accepted the hydrogen. This hydrogen will be released from these carrier molecules in the inner membrane of mitochondria during the oxidative phosphorylation. And this is the stage where oxygen comes into play. And this is the only stage where oxygen is used. Oxygen acts as the final acceptor of hydrogen. Now, we have to keep this in mind to understand anaerobic respiration. We have to keep this in mind that oxygen is really important. It is the final acceptor of hydrogen. It accepts hydrogen, it makes water, which is the second product of respiration. And during this movement of hydrogen, ATP is formed. So this is the complete summary of all these reactions in one diagram. And if we talk about the ATP, the number of ATPs used and made during all these stages, you will see a table here. During glycolysis, 2 ATPs are used for phosphorylation from glucose to fructose phosphate, and then from fructose phosphate to 1,6-fructose bisphosphate, the molecules are being phosphorylated. So 2 ATPs are used. In total, 4 ATPs are made. The net gain is 2. During link reaction, no ATP formed, no ATP used, and the net gain is 0. During Krebs cycle, no ATP used, but 2 ATPs are made. So in total, we have the net gain of 4 up till Krebs cycle. The most productive of these is oxidative phosphorylation. In this process alone, by the breakdown of one glucose molecule, 28 ATPs are formed. So this is really important to understand. Then we have respiration without oxygen. During anaerobic respiration, be it in yeast or in muscles, all the processes should stop actually, but the glycolysis finds an alternative pathway. But the question is, if oxygen comes into play in oxidative phosphorylation only, why would the Krebs cycle stop if there is no oxygen? Why would the link reaction stop if there is no oxygen? So to answer that question, we have to make a bridge. We have to make a link in our minds. Let's understand this. Now, oxygen is the final acceptor of hydrogen. We have understood that. NADs and FADs would release their hydrogen so oxygen could accept it. At the end, in oxidative phosphorylation, all the reduced NADs and reduced FADs that have been formed during glycolysis or link reaction or Krebs cycle, they would release their hydrogens at the end. And this hydrogen will obviously be accepted by oxygen. If there is no oxygen to accept this hydrogen, NAD and FAD would not be able to release this hydrogen. That means NAD and FAD, reduced NAD and reduced FAD, are still reduced. and that means they are already occupied with hydrogen. They cannot accept any more hydrogen. There is no molecule available. There is no acceptor for hydrogen. So that means glucose will not be broken down. Glucose will not be releasing hydrogen because there is no one to accept it. That's why link reaction will stop, Krebs cycle will stop, oxidative phosphorylation obviously will stop, glycolysis should also stop, but glycolysis finds an alternative pathway. That's how anaerobic respiration happens. What's the alternative pathway? If you look at in yeast, the process of glycolysis is same as aerobic respiration. Glucose being broken down into pyruvate. So up till pyruvate, the process is same. But then the pyruvate would release carbon dioxide. When the carbon dioxide is released, pyruvate was a 3-carbon compound. 1 carbon dioxide released or 1 carbon released, it becomes a 2-carbon compound. This is ethanol. And this ethanol actually accepts the hydrogen from reduced NAD to make it NAD, to make it free, to accept more hydrogens. So this process could keep on running. This is happening in yeast, and eventually ethanol converts into ethanol. That's why this process is called alcoholic fermentation. Alcoholic fermentation. Now this alcoholic fermentation is the process that is used by industries to make ethanol. Now let's move on to the process that happens in our body. Anaerobic respiration in muscles or in higher animals, obviously. When the higher animals are exerting too much, when they need high amount of energy, let's say during strenuous exercise, during a sprint, when you need a lot of energy, the oxygen supply is limited because the oxygen has to go into your lungs, then to your bloodstream, then to your cells, then to the mitochondria. So it takes time, obviously. Some of the glucose will be broken down without oxygen. That is anaerobic respiration. So this is what happens here. The process of breaking down glucose into pyruvate is same as aerobic respiration or as the one in yeast. But the pyruvate now directly accepts the hydrogen from reduced NAD to make it free. So it can accept more hydrogens, and this process keeps on running as long as it is required in the body. So this process keeps on running. When the body is exerting too much during strenuous exercise, during any process or any exercise that requires a large amount of energy, some of the glucose will be following this pathway in our muscles. It makes lactate, so this process is called lactic fermentation. Lactic fermentation. And this accumulates in the body, causes fatigue. Later on, it creates the oxygen debt. We have discussed that in our O-level and IGCSE course as well. Now let's talk about the respiratory quotient. In simple words, RQ value or the respiratory quotient is the ratio between moles of carbon dioxide exhaled by moles of oxygen inhaled. So the total number of molecules exhaled during respiration of carbon dioxide and the total number of oxygen molecules inhaled during respiration would be the RQ value. For the carbohydrates, when glucose is being used as the respiratory substrate, you can see in this equation, which is basically the prime equation of respiration, we see that there are 6 moles of oxygen and 6 moles of carbon dioxide. For every 6 moles of oxygen, there would be 6 moles of carbon dioxide. And this value would be 1. But it is not always 1, because carbohydrates are not the only substrate. We can sometimes use lipid as the substrate. So if we use this lipid, oleic acid, we can see that 25.5 moles of oxygen and 18 moles of carbon dioxide. So 18 moles of carbon dioxide would be exhaled, while 25.5 moles of oxygen would be inhaled. The ratio would be 18 to 25.5, and the answer to RQ value is 0.7. Okay. What would be the RQ value for anaerobic respiration in yeast? Well, how much oxygen is being used? 0. How much carbon dioxide is being produced? Well, there is some carbon dioxide being produced. So if something, a number is divided by 0, the value would be infinity. Let's go back to the equation here. So in yeast, yes, you can see that carbon dioxide is being produced, but there is no oxygen being used. So no oxygen divided by, even if there is 1 mole of carbon dioxide. You can use the balance equation, but I'm just giving you an example. Even if there is 1 mole of carbon dioxide being produced, so 1 divided by 0 would be infinity. But in reality, some of the cells of yeast would actually be respiring aerobically. So there is some oxygen being used. So the value is less than infinity. What about if we calculate the RQ value for anaerobic respiration in muscles? It cannot be calculated. There is no oxygen used, there is no carbon dioxide produced. So it cannot be calculated. Okay, moving on. Very important one, especially for your P5. Using Respirometer. And this can also be used to calculate the RQ value, and we will see how. In Respirometer, to one end we have a non-living substance, like it could be glass beads or anything that does not respire, of equal mass, and we have a living organism that can respire, that does respire. And for this process we are using germinating seeds for this particular case, and germinating seeds would respire. But at the bottom we have solution of soda lime to absorb the carbon dioxide. So when the respiration starts, let's say this was the initial value of meniscus. This meniscus was here initially, but as soon as this is connected and the organism that is present on this side would start respiring, it will consume the oxygen, the air from this area. And if the air is consumed, the meniscus will move upwards. So after some time, obviously it will be producing carbon dioxide as well, and carbon dioxide may bring it down again, but that carbon dioxide is absorbed by soda lime at the bottom. So there is no effect on the meniscus due to carbon dioxide. The change is only because of oxygen. The change that we see, the change in the position of meniscus, is only due to oxygen. Okay? So oxygen is inhaled, meniscus will move upwards, the liquid will rise, and we note the change. For a particular time period, let's say 5 minutes, we note the change. Whatever that change is, we call that X. So the difference in the level of meniscus is X for this part. Okay? Now we remove this. We remove the soda lime from the bottom, and this time the germinating seeds would respire. They inhale oxygen, which would make the difference here. Now they exhale carbon dioxide, which would also make the difference here, because there is no soda lime to absorb the carbon dioxide. So we already have the value of X, which was only because of oxygen. Now let's say, and you have to be very careful, let's say the amount of oxygen, let me use the pen here, the amount of oxygen was this. The amount of carbon dioxide is more than oxygen. It is this. In this case, the difference that we have here from the initial value is only this. The difference is called Y. What would be the value of carbon dioxide? X plus Y. Because this is the value of X, which we have already calculated, and this is the value of Y. So the total amount of carbon dioxide will be X plus Y. And if we have to calculate the RQ value for this, the formula would be X plus Y, which is amount of carbon dioxide, divided by X. It gives you the RQ value. And when you find out the RQ value, you can get an idea what the respiratory substrate is. Because when the respiratory substrate is carbohydrate, we know that RQ value should be 1. And if it is less than 1, then it should be some other respiratory substrate, most probably lipids. Okay, let's understand the second scenario. We have understood the first scenario where more carbon dioxide was produced, more carbon dioxide was exhaled in comparison to the amount of oxygen inhaled. In the second one, now let's imagine that this was the value of oxygen, again the same, and we have calculated it. We have called it X. But when the carbon dioxide is exhaled, it's less carbon dioxide. So it comes here. Overall, the meniscus is still rising. Okay, overall, from the initial position, which was this, meniscus comes here. So meniscus is still rising. And this difference is called Z. So we had X, which was the amount of air, amount of oxygen. And now we have Z, which is the difference. How do we calculate carbon dioxide in this case? Obviously, the carbon dioxide, which we have here, is X minus Z. If we subtract Z from the X, it becomes equivalent to the amount of carbon dioxide exhaled. So in this case, if we have to calculate the RQ value, it would be X minus Z upon X. This is how we use respirometer, and it is important to understand the use of respirometer, to understand this diagram, how it works, especially for your P5. Okay, we have already calculated it here. There are two formulas. If there is more carbon dioxide, less oxygen, the formula would be RQ is equal to X plus Y. This is carbon dioxide, and divided by X. More carbon dioxide means X plus Y, because the value of oxygen is X. The value of carbon dioxide is more than that, so you add Y. If there is less carbon dioxide, more oxygen, in that case, RQ value would be, because there is less carbon dioxide, so it is less than X, X minus Z divided by X. This is more carbon dioxide, less oxygen. This is more oxygen, less carbon dioxide. It's very simple. You have to be very clear with the use of respirometer and how it can be used for the calculation of respiratory quotient, as we have seen here. If you come up with any questions, you can write it down in the comment box, and I will try to catch it up with you. Now we will discuss the adaptations of rice, and I'll give you 3 adaptations. Because the rice plant is growing in a flooded land where there is a lot of water, a normal land plant would not be able to grow in this, but the rice plants have adaptations to do so. What are those adaptations? Number 1, as the water level is rising, the rice plants would grow really fast, so at least the tips of the leaves are outside for the diffusion of gases, obviously. And they have large spaces in between their cells, large intercellular spaces. These spaces are for effective diffusion of gases within the cells. Now, for a normal plant, when the cell is not getting enough oxygen, what would happen? Especially the root cells, because the gases would diffuse really slowly in water as compared to air. Obviously, the gases are diffusing slowly in water. Especially the root cells are not getting enough oxygen. So when the cell is not getting enough oxygen, we know that it respires anaerobically. In this case, alcohol is produced. They would do alcoholic fermentation, and ethanol is produced. Ethanol is toxic for the plants if it is accumulated, and that's why there is a high chance that a plant would not be able to survive. But rice plant, on the other hand, has first of all an enzyme called ethanol dehydrogenase to break down this ethanol, and they have higher tolerance of ethanol, so they can live in this condition. Okay, so 3 adaptations. They grow fast when the water level is rising, so at least the tips of the leaves are outside for the exchange of gases with the air. Secondly, they have large intercellular spaces, and these intercellular spaces are called parenchyma. Just like the cells parenchyma in the plants, they are called parenchyma, okay, for the effective diffusion of gases within the body of plant. And the third one is they have ethanol dehydrogenase to break down the ethanol that is produced during alcoholic fermentation, and they have higher level of tolerance for ethanol as compared to other plants, obviously. And this is where we are going to conclude. Make sure to subscribe the channel if you have learned something, share it with your friends, and I'll see you in the next video.